Our next speaker is Xin Liu from BGI. The title of the talk is Recent Progress and Challenges for the 10,000 Plants Project, BGI Shenzhen, China. BGI is an early and important part of the Earth BioGenome Project, building on the expansive vision of BGI that they've had for many years.
Okay, thank you. So I'm setting the timer to make sure that I won't speak too long for that. So today I'm going to talk about the 10kp project. So as mentioned just in the introduction of Harris, you can see that for the plant species, you only have like around 367 out of 3,300 of all the species with genomes available. So it's a great challenge that we will expand it. As you can see from that figure, if you look into details, those green colors indicating the plant genomes with relatively bad assembly statistics comparing to others, if not comparing even to the vertebrate genomes we will have. So speaking of the 10kp project, I will first of all give you some brief idea why we would like to do this. So let's start from the very beginning of the 1kp, which we call it the 1,000 plant transcriptome. Actually, it's not to sequence the whole genome of 1,000 species, but to sequence the transcriptome of a little bit more than 1,000 plant species. So what we aimed at back to them in the 1kp project is to expand what we have in the database, especially like you can see in the NCBI databases, we do not have very clear, we do not have a very detailed data from all these different plant species. And this is what I mean the upper image is what we can get after we sequence the 1,000 of them. So speaking of that, the most important theme of that project is for us to expand what we have for the plant genomes, at least the plant genomic data. So what we finally get, I mean, after more than 8 years of the execution of this project, finally we get this kind of milestone kind of study to include all the data, all the analysis of the 1,000 plant genome data. So what we listed here is several findings of it. I mean, in the major paper we published, we will go to publish, I mean, still unpublished. But you can see that there are, especially to resolve the long-term debate on the clade of different groups of the plants. So data, I mean, this kind of genomic data definitely works very good in this kind of way to help us understand better about the evolution. And speaking about the evolution, this is like the first time we look at this kind of very large scale, especially about how plants evolved, how the flowering plants evolved, how they diversified. So as you can see from here, the gene family expanded, the whole genome, like the whole genome duplication events happen more frequently comparing to other clade, I mean the the other part of the plant species. So this is very important that we can find this kind of evolutionary kind of driving force for the diversification of all the plants. So that is like about the 1kp. But I mean, although the major paper is still unpublished, we are, I mean, in last year, especially during the International Botanical Congress in Shenzhen, so we are thinking of, I mean, the group is trying to think of something that we can make further progress, especially maybe one step forward. So we announced that we would like to sequence 10,000 plant species in the genome, actually comparing to the 1,000 species just in the transcriptome. This is what we announced during that Congress last year, and you can see also quite a lot of reports coming out for that kind of announcement, especially people are trying to know how we can do this kind of work. So back to them, we have this kind of very preliminary kind of project plan that you can see here. First of all, we would like to expand from the previous 1,000 plants to 10,000. And we know that there are quite a lot of benefits, as you can see, for different clade. Definitely there are very important questions to answer. And we can get different kind of benefits out of that, especially for all these plant species, as you know. But we all know that there are challenges, especially, speaking here, comparing to other species, other group of real species. Plants in the genome, they are relatively difficult to sequence. I think you probably know that. So that's the one of the major challenges we think of. And also, we are aware that we need to handle the problems of the sampling, the species identification, funding, and all those kind of issues. We had experiences back in the 1KP project. So yeah, this is what we have in the design when we initiated it. And then we published this kind of perspective in GigaScience this year, describing about all these details, like what kind of species we would like to sequence, especially in different kind of clades. And more importantly, we are trying to set up this kind of workflow that will really make this happen, to sequence that many different species. This is like what we proposed, and we would like to get samples from all over the world. Any people would like to provide us with their samples, especially the samples with very detailed information, this kind of meta information we would like to have. After we execute the 1KP, we know what kind of information are necessary for those kind of samples. So we would like to have the samples over the world, and we would like also to make sure that these samples are definitely with other information, especially for plants. Probably the voucher information, the voucher specimen might be very important for us to consider. If they have that kind of information there, definitely we will consider them in priority. And we designed this kind of workflow, especially for the data sharing. This is also quite important, as you've asked for other projects, and we will discuss during this kind of meeting here these 2 days. We would like to share the data. First of all, it would be shared to the sample providers. After we designed that, after a certain amount of time, we mentioned as 2 months, we will release to the public. And for the further kind of study, we aimed at selecting 20% of them, maybe 20% of those species we sequenced, to be subjected to further kind of analysis. This is also like what we would like to have, especially not to make the data release to be quite late. And then, as the other project, we set up this website, and we are improving it. We have the second version of the website to be released soon. And on this website, you can do this kind of login. You can register to provide your samples. We have all this information for you to fill up there. And then about the technical part, as you've mentioned quite a lot, especially nowadays, about the long rates. So we set up this kind of workflow, especially the technical kind of solutions that we will have. Not just the long rates, but also we would like to consider all the different ways, especially to make it more cost efficient. So as you can see here, first of all, we would like to do some kind of genome survey, quickly check the content of the genome in the genome size, repeat content, those kind of complexity. And then we go through the either the traditional kind of hierarchical sequencing, the currently available, the 10x, or to say they're still in progress. We already have some data for the single tube long fragment sequencing rate we developed, or maybe the third generation long rate sequencing. And finally, we will have the high-C data to make sure that at least many of the, high proportion of the genome we sequenced would be finally to anchor to chromosomes. But we are aware that, as we know, there are quite a lot of plant species with very complex genomes. We are also trying to, for those species, we are also trying to at least have the full-length transcriptomes for those species to let us have better idea, better kind of genomic data for those species. These are the sample requirements we have for now for different kind of strategy. As you can see here, we are given this kind of information to our sample providers in order to make sure that their samples will be processed in a very efficient way, especially about the requirement for the DNA amount and also the continuity of the DNA fragments. And what we have for now as for determining what kind of species to sequence, along with the people working with us, especially to determine the priority list for which we determine more than 10,000 species we would like to sequence. This is to let people know what kind of species we are aiming at. And to prove that we can do this kind of very large-scale kind of sequencing, we do something that's kind of preliminary but proof of concept same. So this is what we sequenced for that. We obtained the samples from one botanical garden, the fresh samples, we make the specimen for that, and we also get all the meta information we would like to have, like the geographical locations and those kind of information taken together. We sampled around 1,000 species, and we identified at least around 300 species. This was also the other problem, that to identify these species really take a long time. I don't know whether that's the case for other clade of the species, but this is definitely the case for plant species. And as that kind of proof of concept, we can see that we generate quite a large amount of data, and we also have very, at least in detailed information from the genome content from this kind of survey. We know better about the genome size, about their evolutionary locations, those kind of relationships, and so on. We'll publish this data soon in GigaScience. And let's come to the overview of the current progress. So we had this kind of plan. I mean, this is mentioned on the website about it. And as you can see here, we aimed at sample around 600 species, sequence 400 of them till the end of this year. And basically, we obtained samples for more than 400 species, I mean, in good kind of progress, and this is what we have sequenced. So for those around 1,000 samples, we surveyed more than 900 of them, and we sequenced, I mean, in the whole genome, I mean, for the genome assembly, at least 90 of them, and we assembled 82 of those species, and you can see for different clades. This is kind of the current progress of that. And then I also have several examples for it. As you can see, the very different, even for the same clade, there are different species with very different genome content there for the algae species, for the fern species, for the margonids, for the begonia genomes. And several examples of it, especially for the begonia genomes, we have sequenced 4 of them. And at least for one of it, we reached to very good genome assembly using the 10x kind of strategy. As you can see here, we finally anchored not so much of so high proportion, but at least more than 85% of the assembly finally to the chromosomes. We obtained the chromosomal level genome assemblies. I think that's majorly this kind of current progress for you to know, and we have the research focus, especially about what kind of analysis, what kind of scientific questions can be answered through those kind of large-scale data sets. As you can see here, photosynthesis, evolution of different clade, like origin of cell walls, and so on. Those kind of research focus is for this project. So about the challenges, as you can know from what I've mentioned, especially from technical part, okay? And we would also like to set up a very a more efficient way to deal with all the sample, to share the data, to do the analysis, and so on. And also funding, data sharing. And this is a very quick summary, and what you can see here, 400 species to be received and sequenced. and at least 80 of them to be assembled, considering about 367 species available with the genome available for plants. I think this is kind of progress we've made. I think time up, and this is a working group. Majorly we start from the previous 1KP project, and people contributing there, the key players there, contribute to this 10KP project. We would like to have their experiences and also all the way they deal with the problems they probably came across previously. And finally, time's up, I know, but I would also like to mention other projects. EBP is quite important for us to better understand each other. For example, here we know that JGI is also like to have this kind of open green genome initiative to anchor the chromosomes. This is also what we probably can form together to this kind of effort to make it more efficient after we have this kind of organization of EBP. I think that is all. They got this template. I will show you probably only this project to show you in this kind of template they provided. Okay, that is all. Thank you very much.
Thank you.